Welcome to your CyberEd CD presentation Meiosis package. Notice, again, we're not talking mitosis here. We're talking meiosis. What follows here is a screen capture of a very old, very is a bit of an exaggeration. It is a CD presentation on meiosis. It is fairly long. And just to be honest here, I don't think I could actually sit through the whole thing myself with undivided attention. So, that being said, there is far more information on meiosis here than you will need. It'll be great stuff if you go on in IB biology and all the rest. You'll already have heard a lot of this. But you certainly will get the basics if you can hang in there for a while. So hang in as long as you can. And then at the end, I'll get back to this lovely little worksheet and go over a few of the answers for you and leave a few for you just to keep you honest.
In sexually reproducing organisms, such as humans, sex cells, namely sperm and eggs, are formed in a special type of cell division called meiosis. This program will explain meiosis and the advantages it imparts to organisms that reproduce sexually. However, to better understand the special processes and divisions of meiosis as they relate to cells, it will help to start with a review of some basic concepts of cell biology. Every living organism is composed of one or more cells. Organisms composed of a single cell are unicellular, whereas multicellular organisms are composed of many cells. You are a multicellular organism. Your body is made up of trillions of cells. Yet at conception, you were just a single cell called a zygote, formed from the union of a sperm cell and an egg cell. Meiosis creates these unique cells, which together combine to form the single-celled zygote, with all of the information required to develop into an entire living organism. Cells that make up different living organisms can be one of two types: prokaryotic or eukaryotic cells. Prokaryotic organisms, such as these bacteria and cyanobacteria, are comprised of a single cell in which the cell's genetic material, DNA, is not enclosed by a membrane. Eukaryotic organisms can be either unicellular, such as yeast cells, or multicellular, like humans. In eukaryotes, the DNA is located within a structure called the nucleus, a region bound by a double membrane called the nuclear envelope. In both types of cells, discrete units of DNA make up what are known as genes. Genes direct the way an organism develops. The molecules it builds to perform life-sustaining reactions, and the characteristic traits you see in each organism. In both prokaryotic and eukaryotic cells, DNA is compacted along with proteins into long strands of thread-like chromatin, which can be further condensed into more compact structures called chromosomes. Different types of organisms have different numbers of chromosomes. For instance, the somatic or body cells of chimpanzees possess 48 chromosomes. Human cells have 46. Pine trees, 24. Dogs, 78. Corn, 20. Frogs, 26. And bacteria have just a single chromosome. Cells often have a duplicate pair of chromosomes, which can be arranged into matching pairs. For example, of the 46 chromosomes found in most of the cells in your body, 23 originally came from your mother's egg cell, and 23 came from your father's sperm. This display of the 46 human chromosomes that have been stained and organized into matching pairs is called a karyotype. The chromosomes that make up each pair are called homologous chromosomes, or homologs. The karyotypes that you see here come from cells of two different humans. The one on the left is from a female, and the one on the right is from a male. At the bottom right corner of each karyotype, you'll notice a distinct set of chromosomes labeled XX or XY. Females have a homologous pair of X chromosomes, whereas males have a non-homologous pair consisting of one X and one Y chromosome. The X and Y chromosomes are known as sex chromosomes because they're responsible for determining the gender or sex of the offspring. All of the other chromosomes are called autosomes. Now you may be wondering, how do the chromosomes get into new cells? Well, all cells come from other pre-existing cells. When cells divide or reproduce, the new cell is typically furnished with all of the chromosomes and thus the genetic material that it needs for survival. The method of cell reproduction differs between prokaryotic and eukaryotic cells. When a prokaryotic cell reproduces, it first makes an identical copy of its DNA and then divides in half to form two new cells, each with the same amount of DNA as the original cell. Since prokaryotes are single-celled organisms, cell division results in the creation of a new organism. This type of reproduction, in which only one parent is involved and the offspring are genetically identical to the parent, is known as asexual reproduction. Eukaryotic cells typically duplicate and divide their genetic material in a process called meiosis. And the rest of the cell contents split into two new cells in a process known as cytokinesis. In unicellular eukaryotes, meiosis and cytokinesis represent asexual reproduction because new organisms are created. However, in multicellular eukaryotes, such as yourself, these processes don't create a new organism, but instead result in new cells that are used for growth or for replacing dead or damaged cells. The processes of meiosis and cytokinesis enable the tiny single cell you were at conception to divide, so that you developed and grew into the human composed of trillions of cells that you are today. Meiosis and cytokinesis take up just a portion of a cell's life, which can be broken into various phases, collectively known as a cell cycle. Cells spend the majority of their time in the stages collectively known as interphase. The cell grows and produces new cellular structures in the G1 phase. During the S phase, the cell doubles its set of chromosomes. Each original chromosome and its duplicate partner are identical, and they are known as sister chromatids. The sister chromatids of each duplicated chromosome are bound to each other by a dense region known as the centromere. During the G2 phase, the cell prepares for the M phase, which is meiosis and cell division. The portion of the M phase, known as meiosis, consists of 4 distinct stages, which you will soon see are very similar to the phases of meiosis. In prophase, the genetic material condenses to the point that individual chromosomes consisting of paired sister chromatids, can be seen under the light microscope. The chromosomes begin to travel toward the middle of the cell, and in metaphase, the chromosomes line up at the equator of the cell. During anaphase, the centromeres that held sister chromatids of each chromosome together separate, and the now individual or daughter chromosomes are pulled toward the cell poles. In telophase, the chromosomes reach the poles and assume a less condensed form. New nuclei form, and the cell eventually divides through cytokinesis. If these processes are not familiar to you, you can review or learn more about them in a textbook or in CyberEd's Meiosis program. It's important to realize the difference between mitosis and the topic of this program, meiosis. Mitosis represents normal cell division. For instance, as an organism grows, the process of mitosis duplicates the genetic material and distributes it equally to each new cell created. Meiosis, on the other hand, represents a very different type of cell division. Meiosis produces new cells with exactly half the number of chromosomes that were present in the parent cell. In meiosis, the chromosomes are duplicated once during interphase, just like in mitosis. However, unlike mitosis, the chromosomes and the cell divide once during the process, called meiosis 1, and once again in meiosis 2, to yield 4 cells with half as much genetic material as the original parent cell. In animals, meiosis produces cells for the sole purpose of sexual reproduction. These specialized sex cells, called gametes, unite with another gamete to produce a zygote, containing exactly the same number of chromosomes as the cells of its parents. Meiosis creates cells that allow sexual reproduction to occur. But what advantages could sexual reproduction have over asexual reproduction? Unicellular organisms reproduce asexually when they divide by mitosis. Many multicellular organisms have a form of asexual reproduction as well. For example, clippings from certain plant species can be planted and will grow into entirely new plants that are genetically identical to the plant from which they were cut. Although this is an efficient means of creating many new individuals, their genetic similarity may pose some serious problems. A single disease or a particularly frosty night could wipe out all of the plants. Whereas in a group of plants with more genetic diversity, some plants may have genes that allow them to resist a disease or survive a frost. In most multicellular organisms, genetic diversity is accomplished through sexual reproduction. Sexual reproduction is the process in which adults produce gametes, such as the human sperm and egg, that both contribute chromosomes to the offspring when they unite at fertilization. The new combinations of chromosomes in each generation create new combinations of genes, and therefore increase genetic diversity. This scene illustrates the human life cycle. In humans, like most animals, egg cells, or ova, produced in the ovaries of the female, are fertilized by the male sperm cells, which are produced in the testes. These sex cells are both produced through meiosis, and each possesses just 23 chromosomes, exactly half the number found in the typical body or somatic cells. The sperm cell and the ovum, each with a single set of chromosomes, are said to be haploid, or 1N. The egg and sperm combine to produce a single-celled zygote containing all 46 chromosomes, 23 from each parent's gamete. Since the zygote contains 2 complete sets of chromosomes, it is said to be diploid, or 2N. The zygote undergoes mitosis to grow and develop into a child, which will eventually become a sexually mature adult. Other sexually reproducing organisms have similar life cycles. The times spent in diploid and haploid stages vary, as you can see by the colored illustrations. Notice that, unlike animals, other types of organisms sometimes have a haploid stage in their life cycle that's multicellular. However, the haploid multicellular stages eventually undergo mitosis to produce haploid gametes, which in turn combine with another gamete to form a diploid zygote. The cycle continues as diploid organisms produce haploid spores, or gametes, through meiosis. If organisms were not capable of undergoing meiosis, sexual reproduction and the formation of gametes from diploid organisms could not occur. You will now learn the steps involved in producing gametes through meiosis. In meiosis, the cell's chromosomes are duplicated in the S phase of interphase, just as they are before a cell undergoes mitosis. Then, the cell goes through the G2 phase and readies itself for the first phase of meiosis 1, which, like mitosis, is called prophase. Prophase of meiosis 1, called prophase 1, is the longest phase of meiosis, typically taking up around 90% of the time that a cell spends in meiosis. In fact, cells can remain in prophase 1 for days, weeks, or in the case of the cells that eventually divide to make human egg cells, years. In addition to the amount of time spent in prophase 1, there are several important differences between prophase 1 of meiosis and prophase of mitosis, which you will learn about in the following scenes. During prophase 1, the nucleus and the dense region called the nucleolus disappear, and the chromosomes condense, like they do in mitosis. However, unlike mitosis, the chromosomes donated by the mother, called maternal chromosomes, shown here in pink, pair up with the homologous chromosomes from the father, called paternal chromosomes, shown here in blue. In males, the non-homologous X and Y sex chromosomes pair at one end. This process of homologous chromosome pairing is called synapsis. When the homologous chromosomes pair during synapsis, the association is called a tetrad, or bivalent. The formation of a tetrad is unique to meiosis and represents one of the major differences between meiosis and mitosis. This photo of cells in prophase 1 shows the thick, condensed chromosomes. Once tetrads form, another process unique to meiosis occurs. This process, called crossing over, occurs when chromatids from the homologous chromosomes actually cross over one another in various places. When this occurs, the chromatids exchange corresponding segments, and they break and recombine, thus exchanging genetic material. The X-shaped region created by the crossover is called a chiasma, or when double or multiple crossovers occur, they are collectively called chiasmata. Earlier, you learned that the combination of a mother's and father's chromosomes that results from sexual reproduction contributes to the genetic diversity of offspring, Crossing over of the chromosomes in prophase results in an even greater amount of genetic variability. These are the reasons that you look and behave differently than any brothers and sisters you might have. The 23 chromosomes that you inherited from each parent were actually a combination of a greater number of chromosomes because crossing over occurred during formation of the gametes that united to create you. With the exception of identical twins, which are formed when a fertilized egg splits, no two people on Earth have ever had exactly the same genes. Near the end of prophase 1, the nuclear envelope of the cell disappears, and in animal cells, centriole pairs move toward opposite poles of the cell, just as in mitosis. Fiber-like microtubules extend and begin to form spindle fibers. The chromosomes, which are grouped into tetrads, remain attached at the chiasmata and begin to migrate to the cell's equator, called the metaphase plate. Following prophase 1 is a very short stage known as metaphase 1, during which time the tetrads migrate and line up along the metaphase plate. Spindle fibers from each cell pole are firmly attached to the homologous chromosome of each tetrad nearest the pole. You'll notice that the tetrads align at the metaphase plate in a random order. The alignment of the tetrads in random order further contributes to genetic diversity of the offspring that will be produced. In this photo, you can see the paired homologous chromosomes aligned at the metaphase plate. The next stage of meiosis 1 is anaphase 1, chiasmata holding each pair of homologous chromosomes together separate, and spindle fibers pull the chromosomes away from the metaphase plate toward each pole. Anaphase 1 of meiosis is quite different than anaphase of mitosis. Notice that in meiosis, homologous pairs of chromosomes separate, but the chromosomes remain as paired sister chromatids joined by their centromere while they travel toward the cell poles. As you may recall, in mitosis, the centromeres holding sister chromatids together split, creating daughter chromosomes, which travel toward the poles. The separation of homologous chromosomes in anaphase 1 of meiosis is called disjunction, which is illustrated in this photo of a cell undergoing anaphase 1. The final stage of meiosis 1 is telophase 1. During telophase, the spindle continues to pull each set of the homologous chromosomes apart until they reach the poles of the cell. In some organisms, the chromosomes revert to their less condensed form. The nucleus and nucleolus reform, and the cytoplasm divides through cytokinesis. In others, the chromosomes remain tightly compacted in separate sides of the cell as it divides in two. The parent cell has now divided into two daughter cells, each containing a set of one of each of the original homologous chromosome pairs. The new cells are haploid, or 1N. But since the previously duplicated chromosomes are still attached as sister chromatids, each chromosome has twice the amount of DNA. This photo of a cell in telophase 1 shows the dividing daughter cells. Following cytokinesis, the two new daughter cells prepare to undergo a second meiotic division, or meiosis 2. Some organisms go directly into the first phase of meiosis 2, whereas in others, a period of interphase called interkinesis occurs. Interkinesis is similar to the normal interphase of the cell cycle, except there's no replication of the chromosomes during interkinesis. Remember, cells in both mitosis and meiosis go through a single phase of chromosome replication, but there's only one round of division in mitosis, whereas in meiosis, the cells undergo 2 rounds of cell division. The cell prepares for this second cell division during meiosis II. Meiosis II starts with a prophase, just like meiosis I and mitosis. In fact, you'll observe that all the stages of meiosis II are nearly identical to stages of mitosis. In prophase II, the nuclear envelope and nucleolus, if present, disappear. A spindle forms, and the chromosomes, which are in the form of paired sister chromatids, migrate toward the equator of each daughter cell. Metaphase II follows prophase II, and like metaphase of meiosis I and mitosis, lasts only a short duration. The spindle is complete, and the chromosomes line up in random order along the metaphase plate in each daughter cell. In anaphase of meiosis II, the sister chromatids of each chromosome separate, becoming new daughter chromosomes. Just as in anaphase of mitosis, the spindle fibers attached to the daughter chromosomes pull them toward opposite poles of the cell. Microtubules that form the rest of the spindle extend and begin to elongate the cell, thereby further separating the chromosomes. This photo shows the separation of the sister chromatids during anaphase II. Telophase II and cytokinesis conclude meiosis II. The chromosomes have migrated to opposite poles of the cell, and the microtubules to which they were attached disappear. New nuclei arise around each set of chromosomes as they assume their less condensed chromatin form. Eventually, the new nuclei are completed. Nucleoli appear, and the cells divide through cytokinesis. Meiosis is now complete. The result is 4 separate haploid cells, with each cell having exactly half as many chromosomes as the original parent cell. It's important to know that each new cell receives a complete haploid set of chromosomes, which are assorted and allocated to the cell randomly. In other words, the cells typically receive a combination of the maternal and paternal chromosomes that existed in the parent cell. Also, since crossing over occurs, many of the chromosomes have a combination of genes from both sets of chromosomes. It should now be clear that meiosis and sexual reproduction lead to genetically diverse offspring. Occasionally, errors in chromosome separation during anaphase of either meiosis I or meiosis II arise. These types of errors, called non-disjunction, occur when pairs of homologous chromosomes fail to separate during anaphase I, or if sister chromatids do not separate during anaphase II. If non-disjunction occurs during meiosis I, 2 of the 4 sex cells produced will carry an extra chromosome, N+1, while the other 2 cells will lack a chromosome, N-1. If non-disjunction occurs during anaphase of meiosis II, the sister chromatids that make up a chromosome fail to separate. In this case, one of the resulting 4 cells will carry an extra chromosome, N+1. One will lack a chromosome N-1, and the other 2 cells will have the normal haploid number of chromosomes, N. A cell carrying an abnormal number of chromosomes is termed aneuploid. If an aneuploid sex cell is involved in fertilization, genetic disorders may occur, and the normal development of the fetus may be disrupted. One example in humans is called Down syndrome, which results from the involvement of an aneuploid cell in fertilization. People with Down syndrome have 47 chromosomes in their somatic cells because they have an extra copy of chromosome 21. Consequently, the affected individual exhibits varying degrees of symptoms, such as stocky stature, heart and circulatory problems, mental retardation, and susceptibility to lung infections. Meiosis describes the cellular processes in which haploid cells are formed from diploid cells. When referring to the production of gametes by meiosis, as it occurs in an organism, the process is called Gametogenesis. In humans and most other animals, Gametogenesis occurs in the testes of males and ovaries of females, forming sperm and ova respectively. The sites of gametogenesis vary widely in other types of organisms. For instance, in flowering plants, the male gametes are found in pollen grains, which are formed in structures called the anthers. The female gametes are formed in structures called gametophytes, located in an ovary. You can learn more about the reproductive processes in plants and other organisms in different CyberEd programs. For the rest of this program, you will learn about gametogenesis in the organisms you're most familiar with, humans. In human males, sperm formation, which is called spermatogenesis, occurs in the seminiferous tubules of the testes. The diploid cells that start the series of divisions that eventually yield haploid sperm are called spermatogonia. Throughout childhood, spermatogonia cells grow and reproduce by mitosis until puberty. Once a male reaches puberty, hormonal changes trigger some of the spermatogonia to divide by mitosis and develop into cells called primary spermatocytes. The primary spermatocytes undergo meiosis I to yield two haploid secondary spermatocytes. Both secondary spermatocytes then undergo meiosis II to form the four haploid cells characteristic of meiotic divisions. These four haploid cells are called spermatids. The spermatids then mature, aided by helper cells called Sertoli cells, to form sperm. Human males continually produce sperm after reaching puberty. You'll notice some distinct similarities and differences between gametogenesis in males and females. In human females, egg cells, or ova, are formed in a process called oogenesis, which takes place in the ovaries. Similar to the spermatogonia that eventually give rise to sperm in males, the original diploid cells from which haploid ova are eventually produced are called oogonia. Unlike male spermatogenesis, however, by the time a female is born, all of her oogonia that will potentially become ova have already differentiated into primary oocytes. These primary oocytes have entered meiosis I, but are suspended in prophase I until puberty. Once a female reaches puberty, a group of cells surrounding a primary oocyte, known as the follicle, will periodically enlarge, and the primary oocyte contained within it will undergo the rest of meiosis I. At the end of meiosis I, the primary oocyte divides unequally to form two haploid cells. The larger cell, which receives nearly all of the cytoplasm, is called the secondary oocyte, and the smaller cell is called the first polar body. The secondary oocyte then begins meiosis II and stops in metaphase II during a process called ovulation, when the follicle ruptures and the cells are released to the oviduct. This cycle happens on average once every 28 days until a stage later in life known as menopause, when a woman ceases monthly ovulation. If a sperm reaches the secondary oocyte, it's triggered to undergo the rest of meiosis II. At the end of meiosis II, the secondary oocyte divides unequally to form two haploid cells, a small second polar body and a large ootid. Occasionally, the first polar body also divides to form two polar bodies. The result of oogenesis, therefore, is four haploid cells, three small polar bodies and a single large ootid, which matures to become the ovum, or egg. The polar bodies eventually degenerate and die, whereas the haploid egg becomes diploid when the sperm nucleus fuses with it, resulting in a zygote, which begins to develop. The rapid divisions in cell growth in a developing organism are nutritionally demanding. These demands are met by the large amount of cytoplasm in the egg. The greater the amount of cytoplasm, the greater the amount of nutrients available for a zygote. The successive unequal divisions of the primary and secondary oocytes form the small polar bodies and furnish the large ootid, which eventually becomes the egg cell, with plenty of cytoplasmic nutrients for the future zygote. This scene summarizes the events of human gametogenesis. Notice the role that meiosis plays in these processes. During meiosis I, the chromosome number in the primary spermatocyte and primary oocyte is reduced from a diploid set of homologous chromosomes to one haploid set of chromosomes, consisting of paired sister chromatids. The secondary spermatocytes and secondary oocytes then undergo meiosis II. In spermatogenesis, the result is four haploid spermatids, which mature to become four sperm cells. In oogenesis, meiosis II also forms four haploid cells, one large ootid, and three small polar bodies. The polar bodies disintegrate, while the ootid matures to become an ovum. Sperm cells and ova each have different combinations of genetic information due to the random assortment of the chromosomes and the crossover exchanges of genetic material between chromatids during meiosis. You now know about meiosis and its role in gamete formation. You've learned three main ways in which meiosis assures genetic diversity in sexually reproducing organisms. First, the chromosomes are randomly distributed to each of the four haploid cells produced. Second, crossing over provides new combinations of genes in the chromosomes. And third, the combining of chromosomes from two different parents provides the offspring with an entirely different combination of genes than those of either parent. As you notice different types of organisms that reproduce sexually, you'll be better able to appreciate the processes that contribute to their diversity and that make each individual unique.
Congratulations on your survival of that. Like I said, way, way, way beyond what you actually need at this grade level, but hey, It's always better to get the full meal deal, and your brain will sort out the rest. So in terms of these actual worksheets that went with the presentation, I am going to go through some of the answers with you here. Make sure you got them down. I honestly don't see the point of you handing this in because, you know what? As long as you know it, that's really what matters here, right? So in your hot seat interview, of course, you'll be expected to show me that you've got it down, some sort of evidence that you've actually made an attempt to learn meiosis. So speaking of meiosis, just what the heck is meiosis? I would say that there's a lot of different definitions, but let's go with a special type of cell division in sex cells. And you know what sex cells are? Egg and sperm. So I'd go with something like a special, that's kind of cool, type of cell division. And again, we're talking about cell division here in sex cells, sperm and egg. What is a zygote? A zygote is essentially just a fertilized egg. Fertilized egg. When Mr. Sperm meets Miss Egg, Mrs. Egg, that's it, much better. You have this fusion of the genetic material in the nucleus of the sperm and the genetic material in the ova of the female egg. That is the zygote. That goes on to develop into an embryo. And if we were talking about humans, of course, that would then develop into, you. What is the purpose of meiotic cell division in eukaryotes? How are prokaryotic cells different in this regard? Well, you are eukaryotic. Your cells are eukaryotic. They're true cells with many organelles, membrane-bound nucleus, etc. So the purpose of the meiotic cell division in eukaryotes is the creating sex cells. What is the purpose of meiosis in prokaryotic? Prokaryotic, by the way, are things like bacteria. They do not have membrane-bound organelles. In a sense, they're simpler cells. They use meiosis, or meiotic cell division, for reproduction. So here where they're actually using it to reproduce asexually. And that's a term you need to know, asexually, without male and female. So what are homologous chromosomes? They are simply chromosomes that have been duplicated. Another way of putting that is a pair of identical chromosomes. Okay, then we have this sweet table. Okay, here you're comparing the total number of chromosomes with how many were contributed by the mother and how many were contributed by the father. So let's look at this. How many chromosomes does a human have? Total number. I think you know. Let's try that again. 46. So how many of those came from mama? Half of 26. 23. How many from papa? 23. How many autosomes? So the difference here between autosomes and sex chromosomes is the autosomes are the regular body cells, the non-sex cells. So we have a pair, of 44 autosomes, 22 of which came from mama and 22 from papa. That leaves, of course, 2 chromosomes, and as you've probably guessed, those are the sex chromosomes. So the question is, how many sex chromosomes does a male have? We have 2. One of them came from my mother, one came from my father. How many sex chromosomes does a female have? Again, 2, and again, they're human, so 1 each. How many whoa, that's a little bit too dramatic here. How many non-homologous chromosomes does a male have? So these are the ones that are not paired. Again, we have a total of 2, and in the case of the mother, 1 would be non-homologous, and the other one would be non-homologous as well. So that, by the way, is a wonderful little animation that shows homologous chromosomes and kind of gives you some of the language involved there, so don't be freaked out by that. I noticed that number 6 isn't actually on your page here, but just in case you wonder, number 6 asks what a karyotype is. Karyotype is simply, it's a picture of an individual's homologous chromosomes that have been stained. The reason they've been stained is so they can actually show up in that picture. Good. I think we should move on to the second page. Okay, the next page shows you 2 karyotypes. Karyotype, like I said, is take any cell of an organism, and every cell, of course, contains the DNA. The DNA has your chromosomes, and in the case of these karyotypes, we're dealing with humans. And how do we know they're humans? We got 22 autosomes. Those are the body cells, and then we have these extra 2 chromosomes. These are the sex chromosomes. So we can tell this one is a female because there's 2 XX, and we can tell this one is a male because of this Y chromosome. So which set of chromosomes belong to a human female and which to a male? So basically you just need to make sure you identify which is which, and then the male would be over here. By the way, there's these wonderful symbols, you know, that you get used to. So the male is basically this guy right here. Then the question, do all the organisms in this diagram have sex cells? The answer is all of them have sex cells because all living organisms contain sex cells. There is one exception, and those, of course, would be bacteria. So these guys down here, these are prokaryotic, and they don't need sex cells. The reason you're undergoing meiosis is to produce sex cells. And whether you're a plant like corn or a pine tree or a human animal, you are going to undergo meiosis to make sex cells. So I guess the answer there is one exception to the rule there. Okay, from scene 7 to 9 in that lovely CD presentation, little mitosis review. Do prokaryotes reproduce by mitosis or meiosis? Technically, it's a kind of a very specialized, well, not specialized, a simpler form of mitosis. If I had to pick one of these, definitely meiosis is about making sex cells, and prokaryotes don't make sex cells. I would be tempted to go with mitosis, but honestly, the best answer here is neither, because bacteria, typical prokaryote, actually reproduces by a simple form of binary fission. They just split in half. Nuclear material goes one way, nuclear material goes the other way, and you have 2 identical cloned bacteria. What kind of reproduction results from mitotic division? I'm thinking here that we are talking not about sexual reproduction, but that other type of reproduction called asexual. What occurs during interphase of mitosis? Well, the truth is a whole lot of things happen during interphase because it's actually a large part of the cell cycle. Technically, we don't even include interphase as part of mitosis. Interphase is taking place during the cell cycle before it enters that mitotic phase. You know, prophase, metaphase, anaphase, telophase, all that good stuff. So the main things that happen during interphase would be the chromosomes duplicating. This is, of course, DNA replication, and a whole lot of cell growth. So if you whoa, what happened there, eh? Somebody wants to give me a lesson someday on just how to work this thing without causing a whole lot of pain to myself and you. It'd be really nice. So the cell grows, and if you remember the cell cycle, there's all those divisions called G1, G2, synthesis phase, etc. But we'll move on. What is the distinction between mitosis and cytokinesis? Well, in mitosis, a cell duplicates and divides its genetic material into two nuclei. So the mitosis part of this is the nuclear division, the nucleus dividing. Nucleus division. Whereas cytokinesis is basically the division of the cytoplasm. Cytoplasm divides into two new cells. Divides. In mitosis, is the parent cell identical to the two daughter cells? Absolutely. Yes. Let's move on to scenes 1 to 11. The following questions are based on human cell division and require some thought and a good knowledge of definitions. Traditionally, these have caused people pain, and given the fact that this video is already much too long, we're going to quickly, we'll not spend a whole lot of time elaborating on these, but if you have any question and need a little bit of further explanation, talk to me. How many paired sister chromatids are there at the start of mitosis? Basically, we have all the chromosomes. They're paired. You have both sister chromatids for each of the chromosomes. So how many pairs? We would have 46. How many paired sister chromatids are there at the start of meiosis? 1. 46. Nothing's changed. How many chromosomes are there per cell after mitosis? Again, mitosis isn't any type of a reduction division like meiosis. Reduction is making simple identical copies. You have a dog with 78 chromosomes. Cells undergo mitosis. You got 78 chromosomes at the end. Whereas, how many chromosomes are there per cell after meiosis is finished? In other words, meiosis 2 goes through 2 phases of meiosis. So there's only going to be the haploid number. So 42 is what we call the diploid number. It's the full duplicate of chromosomes, whereas the haploid number is 23 for humans, because that's really what the point of meiosis was. We wanted sex cells that had half the number of chromosomes. How many autosomes are there per cell after mitosis? Okay, you have 46 to start with, paired chromosomes, but of those 46, 44 are body cells or autosomes. Somatic cells sometimes is what they call them. I'm going to write that. Somatic just means body. How many autosomes are there per cell after meiosis? Now you're going to have half, because you have half as many chromosomes. How many sex chromosomes are there per human sperm cell? Here we go. You've got 2 sex cells, right? If you undergo meiosis, it's either going to be an X or a Y. In either case, that's only 1 sex chromosome. How many sex chromosomes are there per human egg? Again, you're not going to be getting 2, you're going to be getting 1, because it's half as many. How many duplicate chromosomes are there per zygote? Okay, the key word here is duplicate. It can't be duplicate because you're getting 1 from the mother and 1 from the father, so the answer is 0. Tricky. How many homologous chromosomes are there per male zygote? These are chromosomes, homo, same. 44. How many homologous chromosomes are there per female zygote? 46, because her sex chromosomes are homologous. 2 X chromosomes makes it different than the guy, because X and Y are not pairs, right? How many sex cells from mitotic division? Okay, let's read that again. How many cells result from mitotic division of a parent cell? Just 2. And how many cells result from the meiotic division of a parent cell? We have 4. In fact, we have 4. So often here it said 4 haploid daughter cells. Okay, I think we're just about there. People, take a deep breath, and it's all going to end happily. Okay, we are now looking at the end of this. This is a quick summary diagram. You're asked what type of typical body cell has 46 chromosomes. These would be your somatic. It's just a definition of body. Somatic. Where is sperm produced? We're looking at B here, the testes, testicles, plural, 2. Where is the egg produced? Of course, that is the equivalent of the testes in the female. That would be the ovaries. And then we'll move up the page a little bit. What kind of cell division is essential to sexual reproduction? You've got 2 choices. Well, you have more than 2 choices, but if I say mitosis and meiosis, you're going to go for meiosis. But you got to spell it right. Meiosis. The female gamete is the egg, sometimes called the ova, or ovum singular. And that in the diagram up above would be letter E. By the way, these do have corresponding letters here. Somatic cells. The testes were at B. The uterus, or sorry, the ovary is at C. What is the male gamete? Sperm. And are gametes haploid or diploid? Diploid would mean they have the full chromosome number. They have half that, so they are haploid. What must occur for a zygote to form? The only way you get a zygote is if you have egg and sperm come together. That is called fertilization, following a good Christian wedding. Is a zygote haploid or diploid? It is diploid. What kind of cell division accounts for growth and development? That's where your old friend meiosis comes in. The little note at the bottom of the page, label the stages of meiosis 1 and 2 on the attached page and briefly explain what is happening. Given the length of this puppy, I'm thinking I'm going to include that in maybe your first video where we go through the simplified grade 9 version of meiosis, what you actually need to know, and we'll go through the diagram then.